Hello viewers, hope you will find. Today I will teach you about resting membrane potentials. What is resting membrane potential? So resting membrane potential can be defined as, or membrane, this is also called membrane potential. So membrane potentials can be defined as the electrical signals or the basis for processing informations and neuronal response. The impulses which are conducted are generated by the presynaptic and postsynaptic neurons. The resting membrane potentials and the cells, so they are normally more negative than outside. It means that the resting potentials or membrane potentials inside the cells, which are normally more negative inside than outside. This word is from 9 millivolt minus 100 millivolt. This is just the opposite of osmolarity. Excitable tissues of nerves and muscle cells have high potentials than other cells, epithelial cells and connective tissue cells. At this point should be clear that the dead cells do not have any membrane potentials. Whenever the cells they are become dead, so then there will be no generation of membrane potentials or electric potentials. Okay, now let's see here. As we know that one is extracellular fluid, the second one is intracellular fluid. Fluid that is present outside the body, that is extracellular, while one is intracellular fluid which is present inside the cell. Now look at this is the cell. So it's from previous lecture that inside the cell the negativity is high as compared to outside environment. It means that the negativity inside the cell and the as compared to the outside the cell, extracellular fluid, that is greater. Now let's suppose if a cell is polarized because the interior, the intracellular fluid side of the membrane is relatively more negative than the exterior extracellular fluid. Now you can see here this is the cells, and this cell is being polarized because the intracellular fluid, the fluid which is present inside the cells, or you can say the interior, the intracellular fluid, this side of the membrane is relatively more negative as compared to the extracellular fluid, or as compared to the outside environment or exterior extracellular fluid. After that we can say that the membrane potential is due to the sodium ions which are found in the extracellular matrix and the potassium ions found in the intracellular matrix. So this point is clear that whenever the this membrane potentials or action potential, actually this is due to the higher concentrations. This is due to the concentration of sodium ions found in the extracellular matrix and the matrix ions found in the intracellular matrix. Membrane potentials are due to why the membrane potential they are generated. So the membrane potentials is generated due to diffusion of due to movement of ions down their concentration gradients. The electrical charge of ion in any membrane comes from the ions. What is influx? So influx is the net movement of ions into the cell from the extracellular fluid. During influx, the ions move into the cell from the outside environment, from the extracellular fluid, while in efflux it is the net movement of the ions out of the cell to the extracellular fluid. Flux can be defined as flux is the moment of the charges is always measured in millivolts, which is already determined. Now come toward that is membrane potentials. Now come toward the action potentials. Action potentials occur whenever there is a reversal of the normal resting potential going from negative to the positive, which is called depolarizations. So in depolarization, whenever the action potentials they are generated, so there is reversal of the normal resting potentials going from negative environment to the positivity. So that is called depolarizations. Depolarizations, it is occurs when a stimulus causes the voltage gated sodium channels to opens. So whenever the sodium channels, so they are going to opens. So after opening of sodium channels, so depolarization occurs by stimulations. It allows the sodium ions to rapidly influx down its concentration gradient. The sudden inrush of positive sodium ions reverse the membrane potentials for a few seconds. Then the voltage gated potassium channels open. So whenever the voltage gated potassium channel opens, so it can allowing the potassium to rapidly influx due to its concentration gradients. So this brings the membrane back to the negative inside, and this is called repolarizations. So during repolarizations means when the membranes back to the negative inside, mean when the negativity inside the cell it become high. So this process is called repolarizations. As we know that action potential is generated even though the voltage has returned to negative, the membrane is not at resting potential because it is now too much sodium ions inside and not enough potassium ions. The presence of high sodiums inside causes the sodium and potassium pumps to increase by power of 3. The faster is the pump rate, quickly restore the membrane back to its steady state resting conditions. You know, steady state resting conditions when the rate of influx is equal to the rate of efflux. Okay, now let's see here. Here in this case, look at here, these are sodium channels which are open here, and here this channel is closed, which cause inactivations get. It is called open and inactivate very rapidly. Now let's see here, this is the potassium channel which is open here, and this channel is closed, and this is potassiums, and it can open and close slowly. So how repolarization and depolarization occurs? So this is the phenomenon you can see here. So during this case, sodium channel have two gates. This sodium channel have two gates. One is a normal voltage activations. One is the normal voltage activation gate, which is closed at rest. So one is this one's, which is closed at rest, and the second one is inactivation gate. This is called inactivation gate, and this inactivation gate is opens, which is open at rest. So the rapid opening of voltage gate lets the sodium ions rush in and depolarize the cells. We have already discussed that during depolarizations. So during depolarization there is a rapid opening of voltage gates channels, which can allow the sodium ion that can rush inside the cell and depolarize the cell. So when the concentration of sodium ions inside the cell become high, so in that condition you can say that the cell is depolarized, and this is immediately followed by what? This is followed by closing of the inactivation gate, which can stop the sodium influx, and then the sodium ion become unable to go inside the cells. So at the same time the potassium gate channel also open, letting the potassium efflux and repolarization can take place. So during repolarization, the concentration, the negativity inside the cell become high, and the membrane itself become repolarized. So this was all about the membrane potentials and action potential, that how this membrane is activated, and how the sodium channel ions they are opens while the other gates so they are closed. These are their appearance. So you can see here when at home PowerPoint slide check patterns. So you can see here. So all these data have been taken from White's list 2005. Thank you so much for watching the lecture.